Hi, and welcome to the next installment of the AXT webinar series. My name is Cameron Chai, and I'm standing in for my life science colleagues Simon Kinder, Alejandro Rodriguez Kelly, Umesh Nair, and Steve Hunter. In case you're not familiar with AXT, we were established in 1992. We have a diverse product portfolio that spans the life science, material science, mining and resources, and non-destructive testing markets. In terms of our life science product portfolio, we represent a host of manufacturers, including STEM-Biasis, who we'll be hearing from shortly. And we offer cutting-edge solutions for areas including forensics, biomedical research, cell biology, genomics and proteomics, pharmacogenomics, translational research, food and agriculture, and pharmaceuticals. Before we start, I'd like to thank you all for attending, and we'll be more than happy to take any questions that you may have during the webinar. If you have any questions, down the bottom of your screen, you'll see a Q&A icon. So feel free to type any questions in there as the webinar is progressing. So today's webinar is titled Development and Application of Cell-Derived Matrices that Relate to Stem Cell Research. It'll be presented by Dr. Travis Block, who's a senior scientist at STEM-Biasis. He earned a PhD in biomedical engineering from the University of Texas Health, San Antonio, and the University of Texas at San Antonio in biomedical engineering. Currently, Travis serves as a senior scientist for STEM-Biasis and an adjunct professor in the School of Dentistry at the University of Texas Health, San Antonio. So we'd just like to say thanks to Travis for staying back late to give us his presentation, and we look forward to learning more about their product range. So without further ado, I'd like to hand over to Travis, please.
Hi, thank you all for having us and for joining us for this discussion on the development and applications of cell-derived matrices for cell culture. So I'm going to go ahead and share my screen with you guys. So at STEM-Biasis, we are using cell-derived extracellular matrix technology to solve really fundamental problems in cell culture. And when I talk about fundamental problems, I really talk about the very essence of how we culture cells. And so I really like to start my talks by introducing a couple of more famous scientists. And so you may not be familiar with Alexis Carrel and Montrose Burrows, but one of them is a Nobel Prize winner. And they actually published the very first paper on mammalian cell culture back in 1909. And I like to think of these two as the people that ruined cell culture for the rest of us. And so this was the quote. I've translated it for you to make it easier from their 1909 paper. And so bacterial cell culture had been going on for a while from monks making beer, but nobody had been able to culture animal cells. And they were doing microscopy, and they saw cells migrating out of the tissue onto the microscope slides and remaining viable. And so they wrote this paper about how they tried to establish a general method to permit the culture, like that of microbes, of the tissues of adult organs of animals up to man. And oddly enough, the way we culture cells really hasn't evolved a whole lot in the last 100 years. So the way traditional cell culture works is that we take a tissue, we dissect it from the body. That tissue is then either enzymatically digested or mechanically disrupted to isolate cells of interest. And then we take those cells and we put them onto a cell culture dish. And that cell culture dish is often tissue culture treated polystyrene, or it's a dish that's coated in a matrix protein like fibronectin or collagen or laminin or some combination of matrix proteins. But there's something wrong with this system. And the problem is that whatever it is that we're coating this dish in, we're really doing very little to recreate the natural microenvironment from which these cells came. And our philosophy at STEM-Biasis is that you can expect cells to respond just as strongly to signals that they require that they're not receiving as any signals that you do give them. And so when you put cells onto these totally foreign microenvironments, they're going to respond to that. Their phenotype and gene expression is going to change. And sometimes you may be trying to assay a variable, and the cells may be responding more strongly to the bizarre cell culture conditions that are nothing like the body than they are to experimental variables. And so when we talk about cell culture, regenerative medicine, tissue engineering, et cetera, there's really three key components that we're talking about. We're talking about the extracellular matrix, the ECM, the microenvironment. We're talking about cells, and we're talking about soluble factors. And so typically the cells get the most attention because often they are the dependent variable in our experiments, and they are what we are assaying. And the soluble factors get a lot of attention as well, potentially because it's often cheap and easy to manipulate this variable and do iterative testing. Meanwhile, the extracellular matrix often gets totally ignored. And this can be for a variety of reasons. First off, it's complex. So the signals are not just biochemical, but they're also structural and mechanical. And it can be very difficult to manipulate one variable without changing multiple variables in the extracellular matrix. And for a long time, we've had a hard time assaying the extracellular matrix. And so we've really traditionally ignored this variable. But when you look at a cell in its native microenvironment, you can tell that there's just a ton of communication going on between the cell and its matrix. And this has always been really interesting to me because I remember back, going all the way back to high school and middle school science class, being taught that cells are the basic building block of organisms. However, when you think about the number of cells in a human, they're really a relatively small fraction of our mass relative to the extracellular matrix. And so even though we are mostly extracellular matrix, and that is the natural environment in which these cells reside, we tend to totally ignore that variable. And that image is striking, but it gets even more striking when we look at a cross-section of this. So with this image, until it's labeled, it's actually very difficult to tell where the cell ends and the extracellular matrix begins. And that is because the extracellular matrix is interacting directly with the cytoskeleton via ECM adhesion proteins. And so there's just a ton of communication going on that is impossible to discount because the microenvironment is constantly dictating cell fate, and in turn, the cell is elaborating the microenvironment. And this has been starting to change. So over the last 20 years or so, a lot more attention has been given to the role of the microenvironment in regulating cell fate. And on the slide, my favorite part of this here is this study from PNAS in 2010. And what they did here is they took a single substrate. So it was mechanically and biochemically identical in all the conditions. But they made little cookie cutter cutouts so that they could force cells to assume a different morphology. And just by changing the structure of the matrix, they were able to impact gene expression and actually affect differentiation. So what's special about this to me isn't that you force a cell into a shape and it'll differentiate. What's special about this is it shows how difficult it is to dissect the relationships between the cells and their extracellular matrix. And so I like to use this as an example of why it's very difficult to study these cell matrix interactions in a vacuum. And we really need to do a better job of being biomimetic and putting cells in a natural environment. And that's why we view what we're doing at STEMbiosis as really the next evolution in cell culture. So while cell culture has evolved minimally since we discovered it by accident about 100 years ago, or mammalian cell culture anyway, we are trying to really put effort into creating an environment that's more natural so that we can make cell culture more relevant for researchers and hopefully allow them to get results that will translate. And so before I showed you what the traditional model looks like, and this is the paradigm shift that we're trying to introduce. We want to take cells out of a native tissue, isolate those cells, and put them right back into a native environment that they're going to recognize and where they can behave normally and behave like they did in the body. And so the way we do this is we take cells For all of our current products, it's human cells. We put them into a tissue culture dish. We grow them to confluence, and we induce them to secrete a matrix. And once they've secreted that matrix, we gently remove the cells, leaving behind the matrix, and then air dry it onto the dish. So we're able to ship flasks that just look like tissue culture plastic, but they actually have this matrix dried onto the bottom of it so that all the end user has to do is add PBS to rehydrate it. And we have this intact extracellular matrix complete with not just the biochemical cues, but with the structure, the architecture and the mechanics that the cells elaborated. And so sometimes when I describe this, it's hard for people to picture what I'm really talking about. So here we have a really up close, high resolution image of the matrix and the fibrils that it's forming. But just because it's covered in matrix, doesn't mean that you can't see cells on there. It's not going to be opaque. So you can actually image the matrix in bright field. And you can also image both cells and matrix simultaneously in bright field. It also doesn't interfere with fluorescence microscopy. So in this left panel, we actually have matrix stained for collagen 6. And in the right, we have cells on the matrix stained. And so we're able to do both fluorescence imaging of cells on the matrix as well as the matrix itself. And then when we get into the actual function of the matrix and how it's impacting cells, we're actually able to very efficiently isolate primary cells in xeno-free conditions. And so traditionally, very high volumes of fetal bovine serum, very high concentrations are necessary for cell culture. And a lot of times that's because primary cells just really aren't very sticky. They don't want to adhere to these plastic dishes or to fibronectin dishes. And so we add 10, 15, even 20% fetal bovine serum in order to coat the bottoms of these dishes and to make the cells stickier so that they'll adhere. And there are some competitive products out there that claim to be good for isolating primary cells in xeno-free conditions, but in our experience, that just really isn't the case. However, on this matrix, we see that cells immediately home to the matrix, adhere, and begin to proliferate so that we're able to really rapidly and efficiently isolate and expand primary stem and progenitor cells in totally xeno-free conditions. And more importantly, these cells are maintaining a more native phenotype. So I said before that when you put cells into an environment they don't recognize, their gene expression is going to change. And what we see here is a gene expression map. And when you put cells onto a plastic dish, they immediately start upregulating all the extracellular proteins that they're not seeing. And this gene expression doesn't happen in a vacuum. There's tons of crosstalk between these pathways. So when you put cells onto plastic, they start upregulating things they're not normally expressing. And this can be a major driver of spontaneous differentiation and replicative senescence. And this is why we believe that our matrix can help people achieve more biologically relevant results from their experiments. Now on to slide 18, this is actually one of my favorite pieces of data from the company. So across the x-axis, these are different donors of bone marrow MSCs. And on the y-axis, we have their doubling time. And so it's pretty clear that uniformly, cells grew more rapidly on our matrix. But I'm not really so much concerned with the speed at which the cells are growing, because if your cells are growing slowly, you can always just allow them to grow for another day. What I'm more interested in is the variability in proliferation rate. So when we look at cells grown on TCP, there's several outliers. And in labs, both academic and industry, there is this bizarre source of bias, where if you thaw cells and they don't behave exactly the way you want them to in these weird culture conditions, you tend not to thaw them again. So you tend to take those cells, you thaw them, they're not growing fast, you toss those out, you never thaw them again. 5 years later, another grad student comes along, thaws those cells, they don't behave the way he wants them to, and He never thaws them again. 15 years later, somebody asks why their cells from 2003 in the liquid nitrogen tank, and they throw out the whole donor. And so we have this bizarre source of bias where we're only getting results from donors that are already behaving bizarrely well in weird culture conditions. And so on our matrix, by providing cells with a more biologically relevant environment, a more natural environment, we see that we're able to get more uniform responses and more expected responses from more donors. So we can get a better, more biologically representative sample and eliminate a major source of bias. And so most of the data that I've shown you so far was from mesenchymal stem cells, because that's what our original product was originally developed for. However, the first question that we always get is, is this only for mesenchymal stem cells? What other cell types can grow on there? The short answer is it's not only for mesenchymal stem cells, and a lot of cell types can grow on there. We believe, and the data bears it out, that there are a lot of common motifs in various stem and progenitor cell niches. And so by providing this microenvironment, a lot of cells are able to behave more naturally. And so some of those cells, as a non-exhaustive list, have been mesenchymal stem cells from different tissues, human islet cells, some but not all cancer cells. I always hesitate to say definitively anything about how cancer cells are going to respond to our matrix, because by definition, they're cells that were growing in odd places when they shouldn't. Oocytes can improve their viability by culture on our matrix. Bone cells, chondrocytes, renal cells, endothelial cells, monocytes, and macrophages. And additionally, this isn't just for human cells. We've had people growing various rodent cells, equine cells, bovine cells, avian cells, etc. And so in general, we recommend CELLVO matrix for use with adherent cells, especially when you're really interested in more biologically relevant results. And so this first part of my talk has been kind of all about our current products. And then I'll get into a specific application of it. But in summary, for this first half, the CELLVO matrix creates a natural protein-based environment for isolating and expanding adherent cells. The matrix works well with a variety of cell types, improving yields as well as phenotype retention. So we're simultaneously able to get more cells while we're able to get cells that look more like they did in the body. And then we're able to get cells that are more responsive to growth factors when they're cultured on our matrix. We believe this is because they're not busy responding to a bizarre microenvironment. So instead, they're able to respond to your experimental variables. It's important to note that our cells and matrices fit into existing workflows. And so you can use the same kinds of enzymes to attach your cells. You can use your same media. All you're doing is switching out your substrate. And so cells are going to have a more native phenotype when cultured on our matrix. And in short, all of this is about trying to enable more biologically relevant cell culture for our clients. And so along that theme, I'm going to talk about one specific application of our matrix that also leads into a new product that we're launching. And so this is where I might start to sound like I'm contradicting myself a little bit. I just told you that matrices have a lot of common motifs and that our matrix can work well for a lot of cell types. But at STEMbiosis, we're constantly trying to improve the biological relevance of culture. And so we recognize that in our body, there are a lot of different microenvironments, and some of these may be very critical for achieving certain phenotypes. And so one cell type that has traditionally been very difficult to culture in vitro has been articular chondrocytes. So typically, what happens with these chondrocytes is that they start out being kind of round stellate cells. And here we have this de-differentiation process happening gradually, but in reality, it happens very rapidly. These cells, as soon as they start to grow, their gene expression changes, their morphology changes, and their fibrocartilage, or sort of a scar tissue cartilage cell, rather than articular cartilage. and this is marked by a switch from a high call 2, low call 1 to high call 1, low call 2 expression. And these cells, it's osteoarthritis is one of the biggest sources of disability in the world. However, all the research that we're doing on the cells that are involved in cartilage degeneration or regeneration is done on cells that look almost nothing like the cells that are found in our body. And so we set out to try to make a matrix that would better support chondrocyte culture. And so what we did is we took our original product, which was made from bone marrow stem cells, that's the BM-ECM, and then we made an articular chondrocyte-derived ECM. And in these next couple slides, I'm not going to try to read anything into the specific differences that we saw because it's very difficult to tie any one difference in the matrix to an outcome in the cells. But what I want you to get out of these is that cells from a specific source retain tissue-specific qualities, and when they make a matrix, they endow that matrix with tissue-specific properties. And so the bone marrow mesenchymal stem cells and the articular chondrocytes make matrices that have distinct topography and mechanics, and they also have some differences in their biochemical composition that make sense. There's some proteins that are found in one but not the other that we would expect to find in one but not the other. But we're only really concerned with this if it causes a difference in cell behavior. And so when we look at proliferation of the cells, we actually see that either our original CELLVO matrix or the articular cartilage-derived extracellular matrix both supported very rapid proliferation of these human chondrocytes. And so we were able to achieve 400 to 500-fold expansion in roughly 2 weeks on either of our matrices. Whereas on tissue culture plastic, to get less expansion, it took almost twice as long. And so these cells are really growing much, much slower. And their morphology is changing drastically. So these articular chondrocytes, after they've expanded about 400-fold, or 500-fold, I guess, are maintaining a morphology that looks like they are in the body. They have this, they're not very elongated, they have a stellate morphology, whereas on TCP, these are very fibroblastic cells. And so they're getting very elongated and really cease to look anything like chondrocytes do. And this, unsurprisingly, corresponds with changes in gene expression. So on the articular cartilage-derived ECM, as well as the CELLVO matrix, we're able to maintain much higher collagen 2 to collagen 1 expression relative to tissue culture plastic. Not only that, but when we take these cells and put them into a pellet culture to produce a cartilage pellet, we get this very dense pellet that has high collagen 2 expression when cells are pre-expanded on the articular cartilage-derived ECMs. These cells that are cultured on the chondrocyte-specific matrix maintain chondrogenic potential, whereas cells cultured on TCP lost that chondrogenic potential very rapidly. And in addition to seeing that in the collagen 2 expression in the matrix, we also see that the cells that were pre-expanded on the chondrocyte-specific extracellular matrix also had rich glycosaminoglycan expression, which we would expect to see in chondrocytes. And so kind of the takeaway here is that when we take these two cells, they're capable of producing tissue-specific matrices. And then when we take new chondrocytes and seed them onto either the CELLVO matrix or the tissue-specific matrix versus TCP, cells grow well on both matrices, but cells that are pre-expanded on a tissue-specific matrix maintain that chondrogenic potential much better on the tissue-specific microenvironment. And so this is not only key for the therapeutic potential of these cells and for showing that we're able to maintain a biologically relevant culture, but this is also is demonstrative of one of the key properties of our cell products. So when we sell cells, we always sell cells that have never touched plastic. We isolate and expand our cells on tissue culture plastic so that we're able to maintain more biologically relevant phenotypes. Sometimes people are selling cells that have been highly passaged on tissue culture plastic, and they've had time to drastically change their gene expression and spontaneously differentiate so that the cells that you're buying look nothing like the cells you're hoping to study. And in fact, with our human chondrocytes, we sell both P0, so freshly isolated cells, and P1 cells, so that have been through just a single passage, so that we're able to sell chondrocytes that still look like they do in the body, whereas our competitors don't even make any claims about the phenotype of their cells because the cells look nothing like what they did in the body. And so this work about the chondrocytes isolated on our matrix, as well as about the development and manufacture of our matrix, was published earlier this year in Acta Biomaterialia. And this represents also an ongoing project in our lab. And so some of the questions and next steps that we're dealing with now is, on our matrix, we have to wonder about, are the same markers that people traditionally use still the appropriate markers when you're studying them in biologically relevant conditions? And does the improved chondrogenic phenotype translate to improved cartilage formation? So I showed you some evidence that it does, but we'll also need to look at this in a disease model. We're continuing to look at additional chondrocyte markers to further validate this work. And then looking at other aspects of cell culture that may affect the chondrogenic potential outside of the substrate and the population doubling level. And so we have ongoing work that's focused on answering these questions and optimizing our manufacturing procedures to ensure that we're able to deliver a consistent product. But we've had this work ongoing for about 2 years now, and we expect this to be a commercially available product actually later this month. So we are manufacturing a batch right now, and so we're really excited to launch this product. And we think that this work is really a great proof of concept for the value that tissue-specific matrices can bring to research and the value of biologically relevant culture conditions. And so in summary, the ECM produced by either the bone marrow MSCs or the articular chondrocytes have tissue-specific structure and composition and function. And when we controlled for population doubling level of the cells, the articular chondrocytes that were cultured on the tissue-specific ECM maintain a much higher Col2 to Col1 ratio. So again, that's indicative of a chondrogenic phenotype relative to the bone marrow ECM or tissue culture plastic. And this seems to be largely driven by maintaining a low Col1 expression. Additionally, chondrocytes expanded on the tissue-specific matrix, produced more cartilage-like matrix during subsequent pellet culture. And so this tissue-specific matrix may represent an ideal substrate for the expansion of chondrocytes and autologous chondrocyte implantation procedures, and for study of in vitro chondrocyte biology. So we want to be able to probe the biology of these cells that are leading to major sources of disability, but traditional cell culture methods are totally inadequate. And so I'd like to thank the people that contributed to this kind of case study, if you will, on chondrocytes and tissue-specific matrices for that. So we have our team at STEM Biosys and R&D, the group at the New Jersey Center for Biomaterials led by Dr. Joachim Cohn. I'd like to especially thank Dr. Young Mao. And then the lab where I did my PhD work with Dr. Xiaodong Chen, they helped us out with some of the cell phenotyping on the chondrocytes and some of the gene expression work. And so these that I've highlighted played a particularly big role in the publication of the manuscript that I mentioned earlier. And with that, move on to some of the products that were currently offered for sale by STEM Biosys. And so our first product was the CELLVO matrix, which again was derived from bone marrow MSCs. And over the last year, we've added a totally xeno-free version of the matrix. So if you're looking for translational potential in your research and really want to get to cells that are as xeno-free as possible and studying them in as biologically relevant conditions as possible, we're able to totally eliminate fetal bovine serum from the process. All of our mesenchymal stem cell products are isolated in totally xeno-free conditions on our xeno-free matrix. And then we have cord blood endothelial progenitor cells. These are precursor cells for things like HUVEX and HIEX. These are highly proliferative, highly angiogenic cells. And then our chondrocytes that I spoke about a little bit. We are the only supplier that I'm aware of that offers P0 and P1 chondrocytes that are in stock all the time. And so we're able to supply people with cells that look more like the cells in the body. And with all of our cells, it's important to note that they've never touched plastic. They all come from healthy tissue, and they are all very early passage cells. So all of our MSCs are sold at passage 2. And again, the chondrocytes are P0 or P1. Our endothelial progenitor cells are P3. And then we just recently launched our first media products, CELLVO Complete Media and CELLVO Xeno-Free Complete Media. So I mentioned that our chondrocyte product, our chondrocyte matrix, is getting ready to launch later this month, but I also wanted to make you all aware of another tissue-specific matrix that's coming soon. And so this is going to be CELLVO Matrix Plus, and this is a matrix that was designed for pluripotent cell expansion. So some of the features of this matrix are that, like our original product, this is an intact matrix produced by human cells in order to provide structural, mechanical, and biochemical cues necessary for biologically relevant cell culture. It has comparable efficacy to competitive technologies for self-renewal of pluripotent cells, but it's much easier to use. So while some of these competitive technologies require you to do coating steps, some of them require you to coat way in advance, some of them you have to keep the material cold and put your tips in the freezer and put your pipettes in the freezer before you start using them. And that can make it very difficult to coat high throughput plates. With this, again, the matrix is just dried onto the plate. So all the end user has to do is add PBS or media to rehydrate the matrix prior to use. It fits into your existing workflow. So customers do not have to change anything else. They can still use the same enzymes to attach the cells, continue to use their same media. All we're doing is providing a more biologically relevant, easier to use, more effective substrate to improve the relevance of their research. And while this matrix is comparable for the self-renewal of pluripotent cells, it's important to note that we're actually have accumulating evidence that our CELLVO Matrix Plus supports more mature phenotypes of the differentiated progeny from stem cells. And so we're going to be publishing that data soon, but we're really excited to see that we're simultaneously able to support self-renewal of these cells while also better supporting the differentiation, because this has been a major problem in pluripotent cell research where the dogma has been that you're able to make any type of cell from pluripotent stem cells. However, in reality, people have been able to make any type of cell-ish. We get cells that look like neurons. We get cells that look sort of like cardiomyocytes. We get cells that look sort of like hepatocytes, but we're not getting mature phenotypes from almost anything. And so we're hoping that we can really change that with our new product. And with that, I would like to thank our distributors, especially AXT, for hosting this webinar. And I'll open it up to questions. If you don't see a distributor here for you, you're free to contact us directly and we can put you in touch with the right people. And thanks again to AXT. And again, Symbiosis, we're all about making cell culture relevant. And so we think that it's due. With that, I'll take questions.
So Travis, we had somebody ask from the floor, are microenvironments that reflect other specific tissues being developed?
Yeah, absolutely. And so that's also constantly an area of collaboration for us, and we're always interested in hearing about what customers are looking for. I don't want to get too into things that are way far down the pipeline, but some of the things that we're looking at now are substrates for cardiac differentiation and cardiomyocyte culture, as well as neuronal culture. And so we're targeting some of the spaces where people have really had a hard time maintaining native phenotypes of cells. And so we're really excited by the results that we're getting in both of those spaces.
Fantastic. Another question has just come up. We've had someone ask, so do you have different types of matrices depending on the original cells, or are they all generated by bone marrow cells?
Oh, all the tissue-specific matrices are made by different cell types. And so, that's one of the major difficulties with a technology like this, is that, for instance, if you're trying to make a neuronal matrix, neurons are already difficult to culture. We're trying to solve the problems with neuronal culture, so you can't have, a cortical neuron elaborate a matrix for you, at least not in a way that's going to be large-scale manufacturable. So we have to identify cell types that are going to elaborate a matrix that's capable of supporting those cells. And so that's some of the early stage R&D that we do here, and then we move on to optimizing those conditions in order to make reproducible matrices. And so yes, all of the tissue-specific matrices are made from different cell types. And again, we're always excited to hear about what substrates people are interested in. But some of the work that we're focused on right now is in the cardiac and neuronal space, with some early stuff that we're considering in some other spaces.
Fantastic. And just out of interest for everybody else, what plate formats is the cell matrix available in?
Great question. So currently we sell it in 6-well plates, T75s, and T150s. We're actually hoping to launch in October our 96-well plates, and we're getting into other multi-well formats. We're exploring 384-well plates, but there's other technical hurdles that we'll have to work out there. But we're also, in specific cases, open to doing other formats. And so, but yeah, currently we have 6-well plates, T75s, and T150s that we maintain an inventory of, and we'll be launching the 96-well plates soon.
So if there's something that people need, they should come and speak to you guys, and you can probably help them out then.
We do our best to please. But yeah.
Okay, one last question from me. What's the cell matrix made up of?
Yeah, so the matrix itself is actually very complex. And so we don't keep secrets about its composition. Most of that is published either in manuscripts that we've published or in patents. But it's too long a list to go through here because it's well over 100 distinct proteins. Depending on how you do the proteomics, you'll get anywhere between 150 and 3,000 different proteins that are there. And what's been really interesting to us as we've seen some work done on this with our collaborators is that we were hoping originally that we'd be able to take just a few top proteins and mix them together and recreate the results from our matrix. But what we found is that even some very minor components are absolutely critical to achieving normal cell function. And so when we see groups that are, just mixing together a few proteins and saying, hey, there's laminin in this tissue, so let's grow cells on laminin, these cells are expecting to get dozens, if not hundreds, of unique biochemical cues from different components. And those simple substrates aren't recreating that at all. And so we believe that we're actually, one might think that, by adding in all these components, we're adding variables. We actually think we're taking away variables by making it more biologically relevant and allowing cells to receive the cues they do in the body so that they can respond more normally.
Fantastic. So I think that's, for me, that was a big takeaway. It's more that you're creating more biologically relevant environments for the
Absolutely.
stem cells. All right, so just quickly, any last questions before we finish up for today? All right, well, if there's no further questions, thanks everybody for attending, and thank you very much to Travis for taking time out and staying back in your evening to present to us. And
Yeah, thank
in
you.
the meantime, if anybody does come up with any questions, feel free to either email AXT as STEM Biases as Distributor in Australia, or get in contact with STEM Biases directly.
Yeah, thank you all, and thank you again to AXT for hosting.
All right, thanks very much. Bye-bye.
Bye.